A very good morning to all. Dear student, welcome back to the biology class. Today in this class we will continue our lesson 7, that is cell structure and function. Today we will learn about how to prepare a slide to view a cell. Before we have discussed about the discovery of cell, variation in cell numbers and what is cell. So today we will discuss about the preparation of a slide for view cells and other things of the cell, like structure of a cell and what organelles are present in the cell inside. Okay, so all of you be ready with your book. Preparing a slide to view cells. As most living cells are microscopic in nature, they are viewed with the help of a compound microscope. So all of you know that all the living cells or all the cells in the living organisms are what? Microscopic. Microscopic means what? We cannot see them through our naked eye. Only with the help of a compound microscope it is able to view or see their structures, their shape and size. To view a cell under the microscope, the specimen is first placed on a glass slide. This step is called mounting. Specimen means what? The sample. Okay, specimen means the sample. Whatever the sample we are taking. Sometime what? Maybe if we are sick, then that laboratories they used to take lab. So they used to take a blood sample, little drop of blood from our body, and they used to test that. So that is called the sample. Means whatever things they are taking for tasting, it's called the, or they are taking for view under the microscope are called as a sample, and that is called the specimen. Okay. So the specimen is first placed on a glass slide. One glass slide, no one. Here backside it is given. I will show you. Look, this is one piece of glass, triangle side, one piece of glass. It is called the glass slide. Okay, so first the sample will be placed on the glass slide. This type sample will be placed here and that is called the mounting. Mounting can be carried out in the laboratory in two ways. Mounting can be carried out. Mounting are of two ways. They are taking the mounting. They are doing the mounting. Dry mounting or wet mounting. One is the dry and one is the wet mounting. Dry mounting does not require water, whereas wet mounting require water. Dry mount is generally used for viewing inanimate objects. Means inanimate object means inorganic or the organs which are not natural. Like materials and all. Specimens to be viewed under a microscope are often stained using dyes like methylene blue, saffron or crystal violet. So specimen, whatever the specimens are taken, that specimens often stained. Stained means colored. Okay, dye, like we are colored the cloths and also we are using the dye, no? So for mounting or for the specimen, for making a slide also there is some dyes. One is the methylene blue, saffron or crystal violet. These three can used. Okay, the stain are absorbed by the tissue and organelles present in the cell. So whatever the stains you will give, that stains are absorbed by the tissue or organelles which are present in the cell, which brighten, which make the cell inside whatever is there, it's make the everything dark, bright, and differentiate them. If it will be bright, then everything will be differentiated, everything will be clear, making it easier for us to view the detailed structure. So it's make us everything too easy for us to study the detailed structure of a cell. Now here like this, suppose onion peel is taking, So first onion peel is taking. So all of you read this activity properly. Okay. Here given, just I am saying, one onion is taking. Onion is open the take out this outer cover of the onion and taking a inside one peel. It's called a light white color part is there. That one their skin is there. It will take out from the forceps. Then it's placing in a one petri dish. It's called the one plates. Then they use some stain here. They drop some stain here. After that they placed in the this one slide. Then they used to cover with a cover slip. Okay, like that they are doing. After that they will observe under the microscope. Now, structure of cell. A cell has both living and non-living parts. Cell also, even in the living organism, cell are having both the living and non-living parts. The living parts present in cells have a definite shape, structure and function. Whatever the living parts are there in the cell, they have a definite shape, structure and function, and are commonly called as organelles, and they are commonly called as the one organelles. Typically, there are three major parts seen in all cells except bacteria. So, generally, three major parts are seen in the cell except the bacteria cell. Okay, these are cell membrane, cytoplasm and nucleus. In every cell, the cell membrane, cytoplasm and nucleus are present. Clear? Except a bacteria cell. The plant cells have another part called the cell wall, which surrounds the cell membrane, and it is not found in animal cell. So, the plant cell, they are having the another one part that is called the cell wall, which are surrounded by cell membrane, and this is not found in an animal cell. Here the structures of both the cells are given. Look at that structure. This is a plant cell and this is an animal cell. In the plant cell, you have seen one dark, thick cover. Okay, dark thick cover, and that dark thick cover is a cell wall, and that dark thick cover is absent in the animal cell. This is a cell wall, and here the cell wall is absent. Here this thin, very thin one boundary is there. That is called the cell or plasma membrane. This thin boundary is present in the plant cell also. This one, a thin line. That means plant cell have both that plasma membrane also plus cell wall also. But animal cell having only plasma membrane. Okay. So all of you study, see the structure, compare both the structure, what things are present in the animal cell, what things are present in the plant cell. You compare it. Okay. Now come to here. Observe the above diagram carefully. You will see the cell organelles embedded in the cytoplasm. If you observe both the cell nicely, then you will find that the cell, all the cell organelles are embedded with a cytoplasm. In a cytoplasm, cytoplasm is what? It's a fluids which are present inside the cell. Each organelle present in the cell perform different functions. Each organelle, each means every small organelles which are present in the cell, they are doing a different functions. Let us learn about each of the cell organelles to know their function. Cell membrane. A cell is surrounded by a thin covering called the cell membrane or the plasma membrane. I already told you this is cell membrane or the plasma membrane. So, cell is surrounded by a thin covering called the cell membrane or the plasma membrane. Okay. The cell membrane protect the cell and give it shape. So this cell membranes, it's protect the cell, means inside whatever things are there, it is protecting that things of the cell, and it's giving a particular shape to the cell. It allows only selective materials in and out of the cell. Hence, the cell membrane is also called as selectively permeable membrane. So, this cell membrane allow only the selected particles, only the selected means which is important for the cell, only it is allow only that particles to enter inside the cell, and which one is not required, they will allow to go out of the cell. That's why cell membrane is called as the selectively permeable membrane. It regulates the movement of selected substance, water and minerals in and out of the cell. It regulates all the movement of the selected particles and water and minerals to go in and out of the cell. Cell wall. The cell wall is present only in plant cell. Already I told you cell wall is present only in the plant cell. Animal cell, they don't have a cell wall. This extra layer provides rigidity and protection to the cell. These extra layers which are present in the plant cell will give rigidity, means strong. It's making the strong, tight, okay, and it's protecting the cell. As plants do not have skeletal support, so all the animals, they have a skeletal, means bones they have, no, but plants, they don't have a bones. That's why they have a cell wall to support their body. Okay, the cell wall is completely permeable to all substance. Cell wall is not a selectively permeable, it is completely permeable to all the substance. The cell wall contains a large amount of cellulose. Cell wall is made up of a cellulose. That means large amount of cellulose is present in the cell wall. Next come the nucleus. The nucleus, it's the main part of the brain. The nucleus controls the major function of the cell. Nucleus used to control the major function of the cell. Thus, it's referred to as the brain of the cell. So, nucleus is also called as the brain of the cell. The nucleus is surrounded by a membrane called the nuclear membrane. The nucleus is surrounded by a membrane, a layer, and that is called a nuclear membrane. A semi-solid substance called nucleoplasm is present inside the nucleus. Nucleoplasm is present inside the nucleus. A semi-solid, not a complete solid also, it's little bit liquid type substance which are present inside the nucleus is called as the nucleoplasm. The nucleoplasm contains thread-like substance called chromosome. These thread-like structures, have you seen these thread-like structures are there in the nucleus? So that is called the what? Chromosomes. Okay. Chromosomes carry the hereditary material called genes that are responsible for transferring characters from parents to the offspring. So chromosomes are what? It is responsible for transferring the hereditary material, means hereditary material which if the parents having some character, that character they used to transfer to our body also, and that's called the gene. Through the gene they are transferring the body characters to from the parents to the offspring. When a cell is not in a state of division, they are seen as a network called chromatin network. When cells are not in the condition of a divide further, then they are having we will see networks like many chromosomes are there, then it is called the chromatin network. A non-membrane bound spherical structure called nucleus is also present inside the nucleus. So one non-membrane bound spherical structures are there in the nucleus. It is called the nucleolus. It's also present inside the nucleus. Let's see which one is a nucleolus. This is a dark spot, no, dark blue color, have you seen? So it is called the nucleolus. Cytoplasm. Cytoplasm is a jelly-like fluid that is found within the cell membrane but outside the nucleus. So cytoplasm is what? It is a jelly-like fluid which are present inside the cell but not not outside the nucleus. Okay, sorry, no, within the cell membrane but outside the nucleus. Not inside the nucleus, it is outside the nucleus. Many organelles are embedded in the cytoplasm. Other many organelles are embedded inside the cytoplasm. Some cytoplasmic organelles are discussed below. So now we will discuss some cytoplasmic organelles which are outside of the nucleus, which are present. Okay. Golgi apparatus, Golgi bodies also is called. It is made up of a stack of tube-like structure called vessels, which are responsible for the secretion of chemical substances such as enzymes, hormones and proteins. So this is a tube-like structure which are present in a cytoplasm, and this is Golgi apparatus is responsible for the secretion of chemical substances such as enzymes, hormones and proteins in our body. Let's see where the Golgi apparatus. Golgi vessels. Have you seen? These are the Golgi apparatus. Okay. Have you seen? It's the Golgi vessels here. It's the Golgi apparatus. Clear? Next one, mitochondria. These are small rod-like or spherical structure. It's look like a rod or spherical structure. They oxidize food to provide energy and are therefore called the powerhouse of the cell. So mitochondria is called as the powerhouse of the cell. Why? Because they used to oxidize the food and give us energy, or give energy to the cell. Okay, so that's why mitochondria is called as the powerhouse of the cell. Where is the mitochondria? Here, this is the mitochondria. It's the shape of a mitochondria. Okay. Next one, vacuoles. These are sac-like structure enclosed by a membrane called tonoplast. They store excess water, food and waste inside them. So vacuoles are what? It's a sac-like structure, means bag-like structure. Okay, and enclosed by a membrane called the tonoplast, and they used to store the excess water excess water, food and waste inside them. Vacuoles. See the vacuoles where it is? Here. Have you seen a vacuoles? These are vacuoles. Okay. Next one, lysosome. Lysosome contains chemical substance called enzymes that can digest cell and a variety of intracellular and extracellular materials. So look, lysosomes. It is one organelles which are present in a cell that contains a substance called the enzymes. That enzyme digests the cells. That means what? Suppose some cell got hurt and it's damaged, so it is now useless. So lysosome used to kill that cell. Even lysosome present inside that particular cell, damaged cell, but it's kill the cell oneself. Means where it is present, like suppose we are living in our house, and if our house got damaged, we are completely destroying it, like that. Suppose like you have then mean killing oneself. Okay, like you know that committing suicide. So that means what? Committing suicide means if you are getting hurt, you are feeling sad. Some people used to do like that. If they are feeling hurt, if they are feeling sad, they are committing suicide. So lysosome also doing that. if the cell where they are living, if it will be got damaged or if it got hard, lysosome useless, become useless, then lysosome is to kill that particular one cell. That's why lysosome is also called as the suicide bags of the cell, and they are capable of bursting and destroying cell during emergency. But in the emergency situation, means where the cells are useless, now they don't have any work, then they used to kill them. Okay. Otherwise it is dangerous for the whole body or thing. That's why. Let's see the where is the lysosome here and how it look like. This one. Okay, it's like a bag. Okay, packets like. Then next one is ribosome. These are small granule structures scattered in the cytoplasm and are responsible for protein synthesis. So ribosome is a small granules like structure which are scattered in the cytoplasm, and it is used to synthesis or used to produce protein. Okay. Next one, endoplasmic reticulum. This is a network of tubules which are involved in storage and transportation of substance within the cell. There are two types of endoplasmic reticulum, smooth ear and rough ear. Ear means endoplasmic reticulum. So it is a network of tubules and it is involved in the storage and transport. Like it is a package for transporting, when we are sending something from one place to another, so we used to make the packets. Then we used to, like suppose letter, we want to send a letter from one place to another, then we used to keep the letter in one envelope, then we used to put inside the post office box, no? So this one also like doing endoplasmic reticulum also doing like it is storing the food or storing the energy, and it is making a packets like, then it is transporting the substance from one place of the cell to the another. So these are of two types, smooth ear and rough ear. Rough ear has ribosomes attached to its surface, due to which the surface has a grainy appearance. So in the rough ear, ribosomes are attached with that. That's why it is look like a grainy appearance. Okay. The in smooth ear does not have ribosomes. After finishing this, we will again see the structure of that. Then centrosome. This structure is present only in plant cell. Centrosomes mainly function to initiate and regulate cell division. Centrosomes are present only in the plant cell and it is used to regulate the cell division. Plastids. These are present only in plant cell. Plastids contain certain pigments that have specific function. So plastids are also present only in plant cell, and plastids contain some pigments. According to that, that pigments are different, having some specific function. Depending on the pigment color, plastids are of three kinds. So depending on the color of the pigment, plastids are divided into three parts. First one is chloroplast. This contains a green pigment called chlorophyll, which help plants in making food. So all of you heard about the chlorophyll, all of you know about the chlorophyll. When plants used to prepare the food, in the process of photosynthesis, they require a chlorophyll. So chlorophyll is present inside one organelles, that is called the chloroplast. Chromoplast. This contains the non-green pigments. Here other colors are there but non-green. Green color is not there, which gives colors to flower and fruits. Because of this chromoplast, the fruits and flowers of plants are in different colors. Okay. Leucoplasts. These are colorless and store food in form of carbohydrates, protein and fats. They mostly contain starch. So leucoplast is what? It is a colorless. It stores the food in the form of a carbohydrate, protein and fats, and they are contain mostly starch. Okay. So this much for today's class, In the next class, we will continue our lesson and we will learn more about the cell. But before finishing, here I will give you some home assignment questions. You have to write down the answers and send me in WhatsApp group. Okay. So student, these are your home assignment questions. 5 questions are there. Write down the answer and send me in WhatsApp group. Thank you and have a nice day.